Right. So I think that humans have been living with fish for a long time. So you can see them in a lot of the art objects. But when it comes to experiments on fish, I think it comes down to the human curiosity. So, I think that in the centuries past, people have been playing with fish, keeping them for pet, and eating them, obviously. So when it comes to having the fish into the laboratories, it is nothing new. But more recently, I think that in the past 5 decades, zebrafish has been used more extensively for human research. It is one of the animal models for human diseases being used by the National Institute of Health in the US. So scientists worldwide, in addition to all the American scientists, we actually have been using fish, zebrafish, Madagascar fish, to help us to understand how human developed from an embryo into a full adult. But much more importantly is to look at how diseases develop in humans, and then hopefully find cures for them. So in particular, the fish, even though they don't look like us, they don't breathe air, they don't walk on 2 legs, but they actually share several characteristics with us. First of all is that they are what we call vertebrates. So all of us, we have the spinal cord, and fish, they do have them. But much more importantly is that when it comes to the genetic materials, more than 97% of the genes in the zebrafish that I'm working on, or in the Madagascar that I'm working on, they are actually very similar to human beings. So if there is a very important gene in the human beings, there will be a similar type or even an identical type in the fish. So then you can imagine we can actually study at the genetic level or at individual genes level how diseases are being developed, and hopefully find cures from it. When it comes to fish that we work on, there are 2 types. One is what we call the wild type. So this is what you can catch in the wild, but in Hong Kong we actually buy them from the pet shop. So it is really from Mongkok that we go to the pet shop and buy them. It is one of the ornamental fish that people actually keep in the fish tank for their own entertainment. And then, but more importantly is that we have mutant fish or transgenic fish that we buy from the different parts of the laboratories. So worldwide we have laboratories who develop transgenic fish or mutant fish that's got the genes being deleted, and then the scientists buy them from institutes for research use. So one of the typical example is the Sanger Institute in the United Kingdom. So there are known mutants. It's not like the X-Men, we round them up and kill them. But whenever mutants are being created or mutants are being identified from natural breeding, scientists will publish it so that the world community will know about it, but also they will actually deposit it in this sort of institute like the Sanger Institute. Right. First of all is that all the fish that comes into any laboratories, including mine, they have to go through quarantine because you do not know what bacteria, virus, or even parasites that they have. So typically they will be kept in isolation for about 1 month. That is why we call it a quarantine. And then we observe them, and then we also add something like the malachite green, which is to disinfect all the bacteria or parasites on the surface. And so this is what we normally do. But once they pass through the quarantine, we will put them into separate tanks and tag their birthdays, their genetic species. And for mutants and transgenic fish which have genes being modified or deleted, we will actually have to check when they have babies to make sure that the gene that is of interest is still being passed on to that particular individual. So there is actually a lot of work to be able to keep fish that we call this mutant fish or transgenic fish. I'm not too sure whether you can say that it is interesting, but for the past 7 years, my lab actually has been focusing on learning how the zebrafish regenerate their hearts. So for human beings, if you know any of us suffer a stroke, then some heart cells died, and then that is what we call the myocardial infarction. Actually, the patients, even though they recover, our heart cannot repair that well, and certainly our heart cannot regenerate. Regenerate means that to replace the lost tissue. But you know, for all of that we cannot do, the zebrafish, they can do it very well. So in 2002, a group of scientists in the Harvard Medical School, they published a sensational experiment. Is that they just take the heart, you know, pull it out of the forest, cut 20% of the ventricle of the heart, put it back, and the fish 2 months later, it just regenerates the lost 20%, you know, living happily ever after. And so, you know, it really gave us, the scientific community, something to think about. Is that how come the zebrafish can do it? It is truly amazing that, you know, for heart, which is a very functional organ, never sits still, will be able to actually regenerate the lost tissue in, you know, the zebrafish, but not in us. So we actually have been trying to, you know, join the rest of the community in finding out what makes zebrafish tick, why we cannot do it. So after all these years, I think that we, you know, that there are different scientific knowledge being discovered by other scientists, but in our labs, we think that it is something to do with the inflammation. So whenever there is an injury, we all know that, you know, our skin's got red and all that. So we think that it's something to do with the inflammation. So hopefully we'll get, you know, something more solid and about the mechanism which we have dissected. But what we are trying to do is that we want to rescue this type of situation in another fish. So in parallel, we are working with the Japanese Madaka fish. So it is a fish that you can actually see in the paintings of a lot of the Japanese paintings. And they cannot regenerate their heart. So if we do the same experiment to the Japanese Madaka fish, they will just heal, they will repair, but they will not replace the lost tissue. So this gives us, you know, a very interesting experiment, as you can imagine, which is, you know, is ongoing. Is that if in one fish we know that it can regenerate, and if in another fish we know that it cannot regenerate, so, you know, can we actually pick out the factors or the cells that help the zebrafish regenerate, put it into the Madaka fish and see whether it will help them to regenerate. So this is the current work. I think that it will take us another, you know, maybe 10 years or so to be able to do this experiment. But this transfer is actually, we have trying to develop therapeutics in humans in mind. So first is that if we can help another fish to regenerate, then, you know, whether we can help an animal species like the pig to heal the cardiovascular disease, then maybe pharmaceutical firms will be more, you know, motivated and develop new therapeutics for myocardial infarctions in human beings. I think that comes to whether we should do, whether we should conduct experiments in animals. I think that when you think about how many fish are being eaten each day compared to how many fish are being sacrificed or bred for experiments, there is a different issue here. I think that what we cannot deny is that if we want to have drugs, treatments, devices that can use in human beings sometime, you know, somewhere down the road, we actually have to start doing experiments in organisms first. Otherwise, you will be seeing your loved ones suffering, and still, you know, currently there are so many diseases that we have no clue how to cure it. As the Hong Kong society is becoming an aging society, so all the neurodegenerative diseases are, you know, surfacing. Grannies, you know, grandmothers, grandfathers, they are going gaga, they do not know where they are. And this sort of, you know, even though in Hong Kong we are so modern, we are so, you know, the sophisticated a society, there are still plenty of diseases that we cannot cure. And so, going back to the subject that I'm working on, myocardial infarction, when people, have this disease, it is only one way. Slightly and slowly deteriorate, because the heart, you can imagine the heart being such a powerful organ. If part of it is dead, then the rest of the heart will have to work very hard to compensate for it. And so that is why they are called a cardiac hypertrophy. That is, the heart actually becomes bigger after people have a myocardial infarction. Okay, because the heart just have to work harder. Now imagine that the heart have to work so much harder, then individual cells are getting tired faster. And so, the prognosis or that you can think about, it will not heal, and it's just going to go, slowly. I mean, we do have medicines to help, but it will not recover. It will just, stay at the same level, and you will call it a cure already. I think that that goes to, a lot of the philosophy. Yes, the wild type fish that we buy, they are from the pet shop, so they are being bred for humans, either experimentation or for ornament, and if they don't get sold, they will still die anyway. So that goes for, I think that most of the animals around us, except that the fish that I'm working on, they're only 2 inches long. So, we are not going to eat them, never. I don't think that even, they will be used for cat food. So this is purely ornamental. But for myself, I think that it is really what we can do for humankind that is much more important. And I say the biologists, I think that if we design the experiments well, and then if we're actually getting information out of it, whether it is positive, negative information, we're still, doing it for the betterment of mankind. So yes, it's very self-centered. We only think about humans. But I have spent the first 15 years of my working life after my bachelor degree in the public government hospitals. And when you see the patients, you will think very differently. Yeah. So I can tell you my first experience when I saw the patients. So I just graduated from my bachelor degree, and I worked at the Department of Pathology in the Hong Kong Medical School Queen Mary Hospital. So, it's just like any research assistant work. I was actually working on the duct lymphocytes. But because I work in a clinical department, it is the same unit that actually were doing tissue typing for kidney transplant patients. So for kidney patients, either they go on the dialysis or they have an organ transplant. But to be able to do that, they have to match the donor with the recipients, the immunology part, otherwise you will have problems. And so when I saw, my first time I saw the kidney patients walking into the clinic to be tested, it's not like us. So, in Chinese we have a description is that the skin color looks like soil, dirt. It is exactly like that. So I think that, for patients all over the world, whatever we can do for them, it will be good. And that will be, the experiments I want to share.